I'm Omer. Today I'll be talking how we're using stem cells to build a model of the human cortex in vivo. Although if you look in the program, I have a different title. But at the end of the talk, hopefully those 2 points will come together. But I think that this is just a cool idea. Can we do something like that? And during my postdoc, as I was working on this project, people kept asking me, but why? Why do you want to do something so strange? So hopefully by the end, maybe we'll get an idea on how it can also be a clinical tool. So I'll start with the first slide that I always like to start with, although in this audience, it's even a better slide. So innovations in neurobiological medicine tend to fail financially, and that's a really, really big problem because part of our ability to solve these problems is based on funding. And this is somehow most negatively correlated in neuroscience. You can see it over there. And today I'm going to talk about a part of a neurobiological disorders, let's call them, and I'm going to focus on neurodevelopmental disorders specifically, similar to Williams syndrome. And yeah, I just want to mention, so maybe if we can think about why is this failing? Why did we get that? And that's a relatively old study, 2013, but I don't think that things are dramatically different today. And I can think about 3 reasons for why this is the case. So one reason that is really, really hard to tackle, the brain is much more complex than other systems in our body. And during my PhD, I did a lot of work on trying to understand how the brain works, and it's super complicated. So it's not easy to have a model of how the brain works. So that's one problem that is hard to tackle. The second problem is that we don't fully understand the biology of the disorders. And one reason for that, at least with the innovation that we had, maybe since we started identifying the disorders genetically, is that a lot of the studies were done in mice, and human brain development is far different from that of mice. So that's a second problem, and maybe I'll show you today how we're starting to solve this. And the third problem is that we didn't have smart medications, and maybe at the end we'll go into what smart medications are, but today with innovations in CRISPR and ASOs and things like that, it really helps. So I'm going to start by showing you this cool model. These are brain organoids. And how do we make brain organoids? We can take today a sample from anyone's skin, fibroblasts, primary cells, and then we reprogram them in the dish to go back in time, become stem cells. And then those stem cells can be differentiated into brain regions, into brain-like structures. Those are really early in development brain-like structures, but they're highly useful. And the model is really cool because you can take now these skin samples from any individual, not just someone healthy, but also people that have a diagnosed neurogenetic condition. So these organoids can be really revolutionary, and maybe I'll give you one example of how we're making them in the lab. So from here, we're already starting from stem cells that are circulating. We're growing them in a dish. And then at the right day, we take enough stem cells and we aggregate them into little balls of stem cells. Then we put magic powder that drives them to become a specific brain region. In this particular example, I'm showing how we're making human cortical organoids. So those are patterned and differentiated to become a specific part of the brain, the cortex, which in humans is the biggest part, the most evolutionary advanced part, and probably the part that is associated with most of the disorders. And this is how it looks. These are the little brain organoids at day 0, and then at day 6. And you can immunostain them, and you can find all the right and interesting developing brain cells inside. using immunostaining. If you can even put little electrodes in the organoids, and maybe I can have some water, thanks. You can even put a little electrode, and your organoids your neurons, put a little electrodes in neurons in the organoids, and they can even fire. And they are super useful. In these two examples, we use them to model DiGeorge syndrome by creating organoids from people that have DiGeorge. And on the right, we could even make 3 different organoids. One is a cortical organoid, the second one is a spinal cord organoid, and the third organoid is a muscle organoid. So those are 3 different regions in the body, and they create a cortical spinal motor axis. So we could use that to model how the cortex interacts, and it can even, in this example, it shows how activation in the cortical side can contract the muscle. So these are cool studies. They are really cool and useful, but one problem with organoids living in the incubator, maybe we can think about them as having a locked in the incubator syndrome. So they're just these little small cultures living in the incubator, but we know that brain tissue also communicates with the outside world, and that doesn't happen for organoids in the dish. So that's one problem. Another problem is they don't become vascularized, and that's super important for keeping the cells healthy over time. And the third problem is that something that we have a specific problem with in neuroscience is that the neurons in the organoids, they don't mature well over time. So the way we thought about tackling this wasn't maybe very original, but the idea was, let's start with making our organoids just the same way. Start with stem cells, aggregate them, make them little cortical balls. But then, when the organoids are ready, we'll transplant them. Oops. Oh, it's showing. OK. Once they're ready, we'll transplant them into the cortex or brains of newborn rats. And in the brain of newborn rats, we can see them 2 months after transplantation in this MRI image on the left. And we could do this across multiple stem cell lines. It's super successful. It took some time to optimize the surgery approach, but once it worked, it was very useful. Then the first thing we observed is that once we transplant them, these organoids start growing like crazy. So usually in the dish, there will be 2 millimeters in size approximately for maybe the first half a year or a year, and they usually don't grow much. But once you transplant them, they grow dramatically in size. And one reason for this is that they become vascularized. At least that's what we believe. So this happens in the first few days after transplantation. The rat vasculature starts penetrating and vascularizing the human graft. And later, what we will find is that there's microglia cells coming from the rat, and they will also populate the graft. And lastly, we see really nicely that as human astrocytes from the tissue, and I'll show you what's the composition of the tissue in a second, but human astrocytes start making blood-brain barrier-like structure with the rat endothelial cells. Okay, so next we wanted to see what is the composition of these grafts. I keep telling you that we're trying to make cortex, but is it really cortex? And the way we wanted to tackle this, I have more slides about this, but this was the ultimate proof. We did something called single-cell sequencing. So in this methodology, basically you take single cells, let's say only the human cells from the graft, and you transcriptionally profile them such that you can cluster them to the different groups of cells that they are. And what we find is that we find in the graft the 3 main lineages of the human cortex, where one is the neurons, which you can see on the right. These are the little purple and pink colors. At the top left, we can see astrocytes, and at the bottom left, we can see oligodendrocytes. So these three lineages are the key lineages of the cortex, and we can find them. Then we repeated this, but this time, 5 minutes. Okay, I have 15 more. 15 more minutes that I need. Wait, it says 10 minutes. Okay. Okay, then we repeated this on the in vitro derived organoids, and we find similar populations, but somehow the neuron population is way less mature and less developed. So that's a bad sign. And we also find some aberrant populations that once you grow the organoids in vitro for long periods, I won't get into this. But most importantly is that we found this amazing, striking functional maturation. So on the left is a little neuron that we fill with a dye, and we can look at its morphology, and it's tiny and shrinked and very underdeveloped. On the right is a human neuron that grew in the rat for 8 months, and you can see how large and impressive it is. And those branches are also important for creating all the functional connectivity with neighboring neurons. And also this was true in terms of synaptic activity and intrinsic activity. The neurons can fire more, they become real neurons. And this is one of the complaints for people in the organoids. Oh yeah, these are cool, but they remain babies for forever, even if you grow them for a year. Then we want to see, let's take this cool new tool and try to model a disease. And the disease we chose was Timothy syndrome because there's a point mutation in this disorder that causes a calcium channel deficit, and this calcium channel is super important for the development of dendrites in neurons. And we repeated the experiment. We transplanted organoids, but this time we also transplanted organoids that were derived from Timothy syndrome patients. And only with this transplantation approach, we're able to identify this new beautiful phenotype where the dendrites of the patient neurons, patient-derived neurons, are completely shrinked and contracted. And we could do this across the 3 individuals that gave the cells. So there's 3 different iPSC lines here. These are just more examples. Okay, I still have 3 more minutes. Good. Okay, so now the question that everyone's probably thinking in the back of their head, can these human neurons, do they communicate with the rat? So short answer is yes. But there's 2 sides to it. One side is that we can think about input that goes from the rat brain into the human cells and activate them. And this is what is shown here. You can see this fluorescent changes in the human cells that have been infected with a specific virus. And every time we flicker the whisker the whiskers on the pad of the contralateral side of the rat, you can see that the human cells are firing. So this shows that they are getting specific sensory input from the outside world. So this was one problem to solve. The other problem is that also cortex is very important for controlling the behavior of the animal. So can these human neurons control the behavior of the animal? And it's not so easy to test. If you just activate the human neurons, it's not that the animal will start speaking and tell you, oh yeah, I felt that. So the way we did this, we said, let's use a specific virus that expresses an optogenetic protein such that when we activate it, only the human cells will fire. And then every time we activated the protein, we also gave the animal a reward. And after a few days, giving the reward for activation of the human neurons, we found that once we activate the cells, the animal immediately goes and seeks the reward. So this was a confirmation that the cells are integrating, at least at a level where the animals can sense their activity. So why do this? And I only have one last slide, so this is perfect timing. First, as I said, the most important reason is because it's cool. And I think that this is what basic scientists should do. They shouldn't always think about how this can help treat people, and not because it's wrong, That's also works, but it's super important for basic scientists to do things that they love and not things that they have to do, and things that just interest them, and then from this this can inspire people to do groundbreaking work. So, but we already had these animals, and there was someone in the lab that was working on a cool drug for Timothy syndrome that is an ASO. And Timothy syndrome, actually, the mutation is only expressed in 1 out of 2 isoforms that the gene expresses. So the ASO, this specific ASO, which is an antisense oligonucleotide, just tries to hunt down this isoform and shut it down. And it works in vitro, and many drugs do work in vitro, and that's cool. But we had the animals, and I said, okay, let's try and inject them. So we did it the most Israeli way. We took the rat, and it has a little human transplant in its brain, and we just shot this ASO into the cisterna magna and a CSF of the rat. And 2 weeks later, we found that this SPICE isoform, we were able to reduce it. And now it's an in vivo animal, right? So we could see whether the animal is having like toxicity effects, or were maybe affecting important genes for her behavior, because this gene wasn't specific to humans, and whether it's good enough to transfer biologically and sustain. So this was a very, very promising result. Now it's under review, basically accepted in Nature, so it should be out soon. And yeah, that's my last slide, so thank you. Thanks for the lab, and thanks for everyone for listening. Maybe we have time for questions?
Any questions?
So, if I understand correctly, in your organisms, you have specifically excitatory neurons, but many diseases are associated with inhibition and changes in inhibition. So how can you approach that question?
Great, already approached, not shown here. We're trying different ways, but there's a way to produce an organoid that is similar to the ganglionic eminence, where the interneurons are produced, and now we've transplanted animals with the 2 organoids. So now they have both excitatory cells and inhibitory cells. And strikingly, the inhibitory cells develop to the cell types that we find in the adult, which we never find when we culture them in vitro. Okay.
Thank you very much.
Thank you.